Assalamualaikum and hi to the evaluator. My name is JANAH binti BUANG, student from Bachelor Degree of Science in Agrotechnology, Aquaculture. This video is a slide presentation for my project. Okay, the effect of temperature on breeding performance of pearl gourami, Trichopodus leerii using artificial incubation. Okay, before I start, I would like to thanks to all lecturers, especially Dr. Ivan Ko, for his guide and helps for me to finish this project. Okay, I would like to also apologize if there is unwanted sound present in this video, as I record this video from home. Okay, this is the introduction. Gourami is a sub order of Anabantidae and family from Ostronimidae. Locally in Malaysia, we call sepat mutiara. Okay, this species can naturally found at slow moving and calm water. For example, lake and large river. Okay, they are native from Southeast Asia, East Indonesia, Malaysia, and Thailand. Okay, for my problem statement, here is a global warming. The rise of temperature is the main issue in aquaculture industry. Okay, and there is a little information found on the effect of temperature on breeding performance to these potential species, and very little information found on the production of pearl gourami in Malaysia. Okay, significance of study is to clarify the effect of temperature on breeding performance of pearl gourami, and information will be used for local breeders to boost the production at the optimum temperature. And the objective is to study and identify the impact of tree temperature 28, 30 and 32 degrees Celsius on breeding performance of pearl gourami by evaluating the bubble formation frequency, spawning frequency, and fecundity. Okay, the breeding performance define the ability to the brood to produce young by evaluating this bubble formation and fecundity and spawning frequency. Okay, broodstock selections and sex identification is the first step in the methodology. As you can see here, male produce brown and reddish color with pointed dorsal fin. Unlike female, it has a rounded and curved dorsal fin and dull color. Okay, the brood all were purchased in local breeder at Beta Farm Kuala Terengganu. Here is the experimental design. It is located at freshwater hatchery. Okay, I have used 20 liter breeding aquarium with and 200 liter for water bath, and triplicate in each treatment. Okay, here is the thermometer. Four thermometer were used for temperature 30 and 32. Okay, here the breeding method. In this experiment, I used natural breeding method, and the length and weight of the fish were measured in the initial experiment. Okay, I used Salvinia species for the floating vegetation, and a cup was cut into a half to hold and placed at the surface of the water to hold the vegetation plant. Okay, vegetation floating plant. Okay, adult fish, male and female ratio is 1.1, and fish were acclimatized in the beginning of the experiment. Okay, the water quality management. Pearl gourami is an everyday fish, and they could survive at lower dissolved oxygen condition. So, and the water exchange has been done in the experiment 2 to 3 times a week by siphon the unwanted at the bottom of the aquarium. Okay, for feeding management, I have used a tetrabit to ensure the brood have enough nutrition for breeding. Okay, frequency of feeding was twice a day, morning and night. Okay, this is breeding behavior, which is bubble nest formation. when they're ready to spawn, the male will build a bubble nest. Okay, it release the bubble nest is enveloped in oral mucus and release at the surface and tend to stick together. And after that, after fertilization occurred, the male deposit the egg into floating bubble nest. Okay, as you can see the picture here, the bubble formation before and after. Okay, next, and the breeding performance of bubble formation. Experiment was done 50 days. Okay, in a few days, male has construct bubble nest. Data collect every time the bubble present. Okay, fish is then observed until fertilized egg occurred. Okay, next, fecundity. Fertilization that occurred were collect and transferred into incubation aquarium. The egg that stick with the floating salvinia was scooped carefully out from breeding aquarium using bowl. Okay, and the egg placed into the incubation aquarium and filled with 1000 milliliter of fresh water. Okay, after 24, the egg hatch, aeration was inserted, while a larvae were count manually using 50 milliliter of beaker. So this is the formula to count. Okay, and here is our spawning frequency after while result of fecundity obtained. Data of spawning frequency were collect. Okay, a spawning frequency was count with how many times the fish produced in 50 days. Okay, this is the result for bubble formation. As you can see, descriptive analysis and ANOVA were used in this statistical analysis. Okay, for fecundity shows in temperature 28 degrees Celsius. Okay, and spawning frequency. As a recap, bubble formation frequency show more in 28 degrees Celsius, and here was the frequency of bubble formation occurred in 50 days was 8.67 plus minus 13.32, 2.33 plus minus 1.53, and 1.67 plus minus 2.89 for the temperature of 28, 30 and 32 respectively, with no significant difference in in the frequency of bubble formation. Okay, for result of fecundity, it only occur in 28 and 30 degrees Celsius. Okay, fecundity obtained in temperature 28, estimated 624, 124, 564 and 132 eggs. Okay, and temperature 30 was not able to count due to total mortality of the eggs. Okay, while 30 remain 0 until the end. Okay, and spawning occurred in 28 and 30. Okay, spawning frequency was highest in 28 degree with 1.33 and plus minus 2.31, followed by 30 degrees Celsius with 0.33 plus minus 0.53, and no significant no spawning observed in 32 degree. However, there is no significant difference were observed. Okay, this is the discussion. As bubble formation, it is believed that formation of bubble appear at higher temperature because study proved that nest building occurred and egg laid at temperature range 23 to 26, 29 degrees Celsius. Okay, for fecundity, the bubble after observation, it seems impossible and might damage the quality of the egg if fertilization rate to try to count. Okay, number of fecundity in this experiment. It increase the number of the egg hatch. Okay. The estimated fecundity for temperature 30 degrees Celsius was unable to count as the unfertilized eggs are found clumped to each other. Okay, and spawning frequency, higher temperature for the spawning frequency result only occur at 28 degrees Celsius and 30 degree, and no spawning was observed in 32 degrees Celsius. Okay. A higher temperature to the fish gives impact on the spawning time, even though the mature fish were evaluated and oocyte developed were normal. Okay, for conclusion, they suggest that temperature higher than 30 degrees Celsius may negatively impact spawning and hatching rate. Therefore, we do not recommend a fish farmer to breed pearl gourami at higher temperature than 30 degrees Celsius. This should help the production of pearl gourami in Malaysia, since result from temperature 28 degrees Celsius showed positively spawn. Okay, however, the researcher is recommend to extend the study period and add replication in the experimental design for better result. This is for my reference, and that's all. Thank you.